All right, on to lecture 11.2 on clustering as part of multivariate miscellany in the module on multivariate models. No required readings for class, but there are a few websites on clustering that I found really helpful when thinking about both k-means and k-middoid clustering as well as hierarchical clustering when creating this lecture. So if you want more information, it's very clearly laid out here as well. And so an important part of multivariate statistics is to find groups within our data. And so we want to be able to group our data, find groups that are similar so we can try to find patterns. And so we've already talked about a few ways to do this through ordination, where you have treatment variables and you want to see if your data cluster by those treatment variables. So remember, we had that zooplankton community data, and there were different types of water bodies, lakes, canals, rivers, etc. And we might expect that our communities would group by those different treatment types, right? We might expect the communities to be different. However, what do you do if you have less prior information about where the cluster should be forming? What if you want to let your data determine where you should be finding your clusters? And so we're going to be talking about unsupervised clustering here, where groups are just allowed to form. And we're going to do that with k-means and k-middoid clustering, and hierarchical clustering, and also graph clustering. And so k-means and k-middoid clustering is by far the most common type of clustering, as far as I can tell. And it's an iterative process that attempts to find the shortest distance from all points to the number of group centers that you determine, and that's k. And so this is how the iterative process goes. So it goes step 1, you start with your Y data matrix with only the columns of data you're interested in. And that would be coordinate data in this case. And then you randomly pick k cluster centers out of the number of observations or cells in your data set. So you just, if your k is 5, you select 5 observations, 5 rows, and that becomes a center. And then you assign the rest of your data observations to a group within a group that's closest to that center. And then once you've done that for all of your observations for each group, you replace the selected center that you randomly picked with the arithmetic mean of that cluster. And that's for k-means. And if the process is very similar for k-middoid clustering, except for if you're doing this method, once you have your groups chosen, then you choose the center of the cluster, becomes the center where the distance to all points is minimized. And so then you basically repeat steps 3 and 4 until your cluster stabilizes, and that's how you form your groups. So let's look at a data example. We're going to go back to our metabolomics data set, where rows are the accessions and columns are the metabolite data within tissues of each accession. So we have the first column is our accession, and then each column after that is a known metabolite or an unknown metabolite, as you see in the first column. And so this and all of the other clustering methods we're going to be talking about require coordinate data. So you need to create your distance matrix and then pull out your points, so you actually have coordinates if your data aren't in coordinate form to begin with. And so I'm going to do that with the LiPo data with an NMDS. But you can use PCA, PCOA, you can use any option you want. I'm just gonna do this with NMDS to start sort of switching it up a little bit. And so here we've created an NMDS plot like we did previously. And so what you can do is, using the scores function, you can pull out your NMDS scores, or your basically X and Y coordinates, and plot this by hand. And this is gonna help you in the future. So I just wanna point out how to do this. So you use scores, and that pulls out your NMDS 1 and 2 columns, because we selected 2 dimensions here. And then those are going to be your XY points for all of your data. So then once you've pulled the scores out, you can plot your NMDS points of all your accessions by hand with ggplot. Just looks like a regular old ggplot. And then next you can run your k-means clustering on those points. So what you're going to do is take your data set of all the accessions, and then you're going to bind your scores to them, write your XY coordinates, And we're going to set a seed here because this is an iterative process. So that way, if you run it, you're going to get similar results to me. And so using the k-means function, you select your data set. You first enter your data set, which are is LiPo is the data set, but it's those last two columns for the scores or the XY coordinates. And you specify the number of centers you want. We're going to start with 5. And then you do the number of starts you want. Here I'm choosing 10, but you can choose whatever number you want. And then this is the output. You get a bunch of different things. Really what you want to focus on is clusters. It's that first column, but I'll show you what that looks like. So what's basically happening here is that the k-means is cluster with 5 clusters, and you're getting, within your 5 clusters, you're getting 7 accessions in one, 9 in another, 15 in another, 5 in the next, 4 in the next. And then if you wanted to plot the means, right, because it chooses the arithmetic mean of all those points, you have your 5 mean cluster XY coordinates in case you wanted to plot them for any reason. The clustering vector is the cluster that each accession falls into, and then some various other information that you can get about your k-means output. And so then if you want to plot your NMDS by hand again, you can color them by their clusters, because if you want to look at the code for how to do that, feel free, it's behind the hood, but you basically bind the data sets together and then you can color by their cluster numbers. And so here you can see these are the points that tend to be clustering together based on k-means. And so if you wanted to do this with k-medoids, it's very, very similar function. The code is again behind the hood, but it's basically identical. You just use k-medoids instead of k-means. And so instead of showing that to you again, I'm just showing you the figure. It looks basically identical because the points are so separated here that that's really not surprising. But so k-medoids is quite similar to k-means. Okay, so let's think about next hierarchical clustering. And so hierarchical clustering helps you cluster points into hierarchical groups, and this algorithm repeatedly tries to combine the 2 nearest clusters into a larger cluster until you get down to one single cluster. And so this is again an iterative process, and this process goes: step 1, calculate the distance between each pair of points, and then step 2, you're going to put all your points all points go into their own cluster. And then in step 3, you merge the closest pair of points based on the distance. So we're going to start with which are the 2 closest, and then the next 2 closest, and the next 2 closest, on and on until so I guess you're not repeating step 2, you're just repeating step 3 over and over until you get everything merged into a single cluster. And so this can be really helpful when visualizing this type of clustering as a tree. So each smaller cluster merges until there's a single cluster at the base of the tree. So here, the closest clusters, say, might be the annelids and the arthropods. You might merge those together, and then next you might merge together the echinoderms and the chordates, and then maybe the next ones you would merge would be the nematodes with that annelids arthropods cluster already. And so you just keep clustering them down, keep merging them down until you get to one single tree. Here, the ancestral colonial protists. Okay, so how do you actually run hierarchical clustering? You use the hclust function, and I misspoke earlier when I said that all of our methods that we're going to be talking about use XY coordinates, because actually hierarchical clustering, you need a distance matrix. And so because of this, you want to make sure your data are scaled appropriately. And so because we're going to use our LiPo data set again, and you're going to standardize your data by site totals just to make sure everything is standardized. And then you can specify the linkage method in your hclust with the method equals. And so for that, there's a lot of different options, and I would suggest if your field is doing this, you should look at it, or just look at the help function to figure out which one you think might be best for your purpose. But the nice thing about this one is you can use any distance matrix you want, distance measure you want. And so this is where you can start thinking about things that are more related to genetic distance or actual Euclidean, physical Euclidean distance, or here we're using Canberra because that works pretty well for metabolomic data. And so you use your hclust function on your distance matrix that's scaled. And then you're going to use the average method. And then what you get out is this just very short description of the clustering algorithm. And then you can plot that, and it creates a really nice dendrogram of how related and how the algorithm paired the points. Or excuse me, here it would be your accessions. And so what you can do then is cut the tree. So you can cut the tree to get the desired numbers of clusters you want. So you use the cut tree function and say you want 5 clusters like we had previously with k-means. You would just take your hclust data or output and then specify the number of clusters, and then you get your output of which accession is in which cluster. You can do the same thing where you cut the tree where you want your clusters to be 3, and it finds that level of the tree where we will get 3 clusters out of how things formed. And so here you can see it just goes to 3 in your output for each accession. And then you can bind this column of clusters to your original data set and again plot it like we did for the k-means and k-midoids to look at which of your accessions are in which of your clusters. Okay, so the final thing we're going to be thinking about is graph clustering. And graph clustering uses a graph theoretic framework to select points that are similar based on various criteria. The ones I tend to think about most often are nearest neighbors or some sort of distance cutoff, but I'm sure there are many more. And so for those that don't know a lot about graph theory, as an ecologist, I tend to think about this as networks of habitat patches, and are your habitat patches that are fragmented connected or not? But there are lots and lots of ways to visualize these networks. This is just what I'm going to use for this purpose. But basically what you think is that each point is a node or a patch. So each patch, you can see the little blobs. You can put a centroid in there, and that's going to be that's a node. So each habitat patch here could be a node, but it could be anything, right? You could have your nodes be accessions in your LiPo data set. They could be individual plants, right? So they could be anything, any row of your data set. And these nodes are then connected by edges. So any 2 nodes or here patches that are connected by movement between, for however defined, that's an edge. And so what happens is you create these networks depending on distance between different observations in your data set. And so if you're interested in this, I would, and especially if you're interested in conservation biology or ecology, I would suggest looking at this really nice paper that really defines what network theory is and all of that kind of stuff. You don't have to worry about it too much for this, what everything actually means. So we're going to talk about this using a data set that I collected and when I needed to use actually graph clustering to figure out groups of my points. And so I had plant locations of Artemisia frigida, which is prairie sage, in a prairie. And so I had those points are the location of all individuals within a prairie. And I was trying to sample seeds from these mother plants across the prairie, but I couldn't obviously collect seeds from all of them because it's a really intact, beautiful prairie, and I don't want to collect all the seeds from the whole thing. But I wanted to select plant seeds to be collected from like groups of plants across the prairie in an unbiased way. So I didn't want to just collect, where it looked like plants might be similar. I wanted the computer to tell me where I thought that I should be collecting plants from, so as to sample the entire area. And so I'm going to run you through this. There are lots of ways that you could do this. And again, you're going to need to coordinate data. But this is just one example that I used. And if you want to talk through any of your specific examples, I'd be happy to talk through how I might do this with your data. So the first thing you do is create a distance matrix. And, here we are back to distance matrix. So I used Euclidean distance because there's actually the physical distance between plants. So I created a Euclidean distance matrix, and Actually, this is a tiny bit different than what we've been doing before in that my data set is just you'll see if you look at the data, but the Artemisia data set, columns 7 and 8, those are actually your XY coordinates of all the points. And so this is different than how we've been creating distance matrices in the past, but this will just allow you to create a distance between all sets of points. And so here's just a histogram of the distance between all plants in the prairie. And then what you do is you have to have some kind of cutoff for deciding where you want your clusters to be. So in the past, with the past methods, we said, okay, I want there to be 5 clusters or 3 clusters. But here I'm saying I want some sort of distance cutoff, and then the computer will pick how many clusters that it thinks are the right number based on that distance. And so what I did is I picked a quantile cutoff from the histogram of neighbor distances of 10%. And so basically, if you look back at this last histogram, I said, okay, what's within the first 10% of that distribution? Those are the 10% nearest neighbors to every plant. What's that distance cutoff? And so I use that 10% to create a network, network elements that are all within the closest 10% of distances to each other. So the 10% quantile of that histogram is 29 meters. And so basically 29 meters then became my distance cutoff for all points. And so you can use the iGraph package to run a lot of network model stuff. And iGraph does a lot of stuff. A lot of people have added to it. It's has, for better or worse, it has a lot of interesting things, and it's not always explained that great. So I just want to point out that here I'm using an edge list, which is a data frame that describes the distance between each set of points. To create graph clusters, there are lots of other ways to do it, but this is how I'm doing it. I suggest trying to read the help manual if you think you might have other ideas, or rely on the literature if there are methods that your literature uses. But so first I made a data frame out of this is a little bit hacky, but it works, so bear with me. So I first made a data frame out of the distance matrix. And then I had to make sure that all my rows and columns were labeled with the IDs of the plants so I could keep track of them. So that's what that next set of code is doing. And then basically I had to melt the whole thing so it looked like one long data set with the ID of one prairie, the ID of another prairie, and the distance between them. And I could not show it to you because it was so long that R Markdown wouldn't let me print out any of it for you. So unfortunately, you don't get to see it. But if you run it in your own R, not through the R Markdown where you create a PDF, then you can see it. So then once you've created the appropriate data frame, the next thing you can do is create your graph using the distance cutoff we determined before. So write the nearest 10% of neighbors. And so we're going to be making this graph from an edge list. So we're going to use the graph.edgeList function and then I know this is a bit complicated of syntax, but basically it's taking the df melt that we made, and again I had to use melt instead of any of the tidy stuff because I for some reason just couldn't get it to work, so I stuck with the old melt. So I know that's a little bit old, I apologize. And then basically it's saying, okay, we're going to take that edge list that we had made, df melt, and we're going to remove any values that are greater than or equal to that quantile that we picked. So we basically want anything that's less than those quantiles. And then we want a non-directed graph. Most graphs are non-directed. And then you simplify it to remove your self loops. And then you can print it out, but this isn't really all that helpful, but it's just showing that it's looking at all the edges. So it's looking at the edge from plant 1 to 2 and plant 1 to 3 and 1 to 4, et cetera, et cetera. So then once you have your graph, you can create your clusters. So this is where we're going to create our clusters. And so there again are a lot of ways to create clusters. I'm using cluster fast greedy here because it's quick. And I was tending to get the same results over and over, but I would recommend looking through iGraph to see if there's any type of clustering functions that you might prefer, but this one worked pretty well. And so basically you run the cluster fast greedy on your network that creates the clusters based on that distance from your network. And then you can organize your data set a little bit. And then here you can then pull out what you get from this code is that you have your cluster value and the ID for each plant. And so then when you merge that, again, the code for this is under the hood, but you can merge those clusters in with your data set and then plot them and color them by clusters. And you see here that the computer is predicting that I should, using this network model, that I should get 6 clusters. And, you know, you may or may not be happy with this, so you can sort of mess around. So that pink cluster, cluster 1, is really covering quite a bit of space because there's a lot of plants that are within that 29 meters, and so they're connected enough that they can, that they stay connected. But right, for my purposes, I may say, okay, I'm not really sure that I want a cluster that big, because if I'm selecting this equal number of plants from cluster 1 as cluster 4, 5, that one that's down in the bottom, then, you know, I really wouldn't be sampling the clusters in the same way. And so I may then go back and try, depending on what your purpose is, and I may try to smaller distance cutoff and then see if I get more evenly spaced clusters. So this is just one of the many ways you can do graph clustering, but I found it quite useful in the past. And so hopefully that helps think about graph clustering. And with that, please complete quiz 11.2 on Canvas and continue on to the next lecture.